Thank you, Dr. Loy. Thank you, everyone, for coming. Dr. Huang and I actually share this title, and I will be doing the first half of the presentation, and she will continue on with the second portion. Let us get started. Today we'll speak to you a little bit about biomarker, co-localization, analysis, and quantification. But let me begin by telling you some of the services that are offered through the TRIP laboratory. And TRIP stands for Translational Research Initiatives in Pathology, for those of you who may not be familiar with our name. So the TRIP laboratory, the specific goals include the following: to facilitate translational research that focuses on pathologic basis of human diseases, to provide state-of-the-art services for users of this laboratory, and to become a regional center of excellence for quantitative pathology and molecular testing in human diseases. So today we'll begin very briefly. I'll speak to you about tissue services and imaging that is available through the TRIP laboratory, and then we'll focus our efforts on the Aperio digital slide scanner, which is a new piece of equipment in our laboratory. We'll move on to the nuance imaging, followed by Vectra imaging, and image quantification using Inform software. So TRIP is a unique service because our staff members have a variety of backgrounds in both the clinical and research arenas and can provide technical support for routine morphology-based techniques. The TRIP lab also has a molecular aspect of its services. However, today we're going to focus on the tissue services and imaging aspect of the TRIP laboratory. In addition, pathologists are available that can help guide a project, help you interpret your results, and collaborate on publications when necessary. For those individuals that may not have a microscope or that may not have a camera attached to their microscope in their own laboratory, imaging assistance is available for either presentation or publication as needed. And we strongly encourage investigators that are new to utilizing human tissue or even rodent or animal tissues to meet with individuals within the TRIP lab to help get them started with their project as it pertains to either harvesting their tissue, fixing the tissue, in order to generate the best possible data from those resources. So what can TRIP provide to investigators here on campus? So I'm going to break it down into several different services. For histology, we can take the tissue that you have and process it if they're formalin fixed. If you have frozen tissues or formalin fixed tissues, we can provide tissue slides for you for analysis and confirmation of a diagnosis. We can perform H&E stains or other special staining techniques. Unstained slides for downstream studies can be provided, and we work with other entities on campus for the creation of tissue microarrays for effective and efficient analysis of multiple tissue types in a smaller surface area. We then provide other additional methodologies for investigators, such as immunohistochemistry or immunofluorescence or in situ hybridization. We can do these staining techniques on slides that have been fixed on cells that have been fixed on slides, on single tissue sections, or on TMAs. Immunohistochemistry and immunofluorescence can be performed using commercially available antibodies, or we work with investigators that have developed their own antibodies. Various chromogenic detection systems are available for colorimetric detection or for immunofluorescence, and other hybridization analysis is available, including for microRNAs as well as in situ hybridization. Moving on to microscopy, we can provide histologic and then immunohistochemical, immunofluorescence in situ slide interpretation. We can provide you with digital images using the Aperio slide scanner. These images can be acquired using the nuance system, which I will speak about briefly. Also, these images can be acquired using an automated Vectra system. Once images have been acquired, they can be analyzed using the two software systems here, either Nuance or Inform, And we have consultative services, so study design, slide interpretation, and then data acquisition and interpretation of those results are also available for investigators. So some things for investigators to think about. We can perform many of these modalities on various different platforms. So as I had alluded to previously, cells can be grown as a monolayer on a slide and then subjected to downstream studies, or they can grow them in culture and then pellet or freeze them for additional downstream studies. Tissues can be whole tissues, either from human or animal, and those can be frozen or paraffin embedded, either a single piece of tissue on a slide or as part of a tissue microarray. Things to consider, these won't be discussed in detail here, but just some things for investigators to think about in advance is what types of antibodies will you be using, the fixation method, the type of sectioning that you're interested in, etc. So detection systems, whether they're direct or indirect, whether you're performing fluorescence, and whether you're interested in multiple labeling or multiplex immunohistochemistry or immunofluorescence. And this is something that we'll talk about in the next section. So these are important things to consider because it will affect how you harvest your samples. So we all know that the location of cellular antigens can be in various compartments. So it can be cytoplasmic, it can be located as demonstrated here in the nucleus, or it can be present in the cell membrane. Many investigators are interested in more than one marker, for example, and in the past have utilized multiple tissue sections to stain for those markers. But performing multiplex immunohistochemistry or immunofluorescence will allow you to identify those markers in one tissue section. And so I'd like to touch about why would one want to do multiplex immunohistochemistry or immunofluorescence. So this type of assay expands the power of your biomarker analysis. Multiplexing has many benefits and applications, and so you can determine and quantify the immunoreactivity as it is co-localized within your tissue of interest, whether it be in the same compartment, so whether it's both in the cytoplasm or within the same cell, for example. Evaluation of spatial relationships between these immunoreactive cell types, studying signal transduction pathways. Most importantly, you're reducing the amount of tissue that you're using for these studies by performing multiplex studies. You're decreasing the amount of reagent. And for those of us that are interested in imaging modalities, you're decreasing the time that you're spending at the microscope and at the computer. So at present, with the software and the equipment that we have, we can multiplex approximately 5 to 6 biomarkers or markers in one tissue fragment, and we are able to keep intact morphology, giving us very important spatial information. So as stated before, we're using fewer reagents, and the precious tissue is preserved as a result. So this allows extraction of meaningful data from the tissue, and it maintains it in its native microenvironment. And the results are more consistent since they do not rely on visual interpretation, but instead benefit from machine-based algorithms, so the computer-based algorithm. So here we have a tissue fragment, and we can see that in this particular image, there is a brown stain and a red stain. For those of you that are performing prostate cancer research, this image is probably something that you've seen quite often. Additionally, here is a multiplex image that shows at least 3 different marker types stained in one tissue section. For those investigators that do immunofluorescence, this is just a nice image for you to see that you can have membranous staining, cytoplasmic staining, and nuclear staining all within one image. So what are some current applications of this multiplex modality? So individuals are using it for biomarker discovery, validation, and analysis, identification of signal transduction pathways, prognostic studies and predictive tests, drug response, very hot area here within our cancer center, cancer research, vessel counting, Gleason pattern assessment, transplant rejection, and stem cell research are just a few areas where this type of technology is being utilized. So before we get into the imaging modalities, we have a new piece of equipment within the TRIP laboratory, and this is the Apereo ScanScope digital slide scanner. And so what this is, it's a rapid and seamless platform that allows you to image whole slides. It's automated. It can capture the image within minutes. It's automated at present for up to 5 slides, and they can acquire these images at 20 or 40x magnification. This is an ideal tool for those investigators that want to capture their entire slide and send to their collaborators or use for educational purposes. There is free software that allows you to view and edit these slides without waiting for image processing. And again, you can share these with your collaborators and with your peers. So some of the things that the Apereo digital image will allow you to do is you can rotate an image. It allows you to edit and annotate images. So within the image you have, you can take a small snapshot of a smaller area of the entire slide. You can annotate on there, and that's demonstrated with circles, notes, measurements, etc. It will allow you to open numerous windows. It will allow you to synchronize multiple slides together. And this is a very nice imaging, just a visual imaging tool. And so here we have a screenshot of what typically can be seen with the Apereo slide scanner. And so up here in the upper left-hand corner, we have the scanned entire slide. On the right, we have an area that has been zoomed in from within that whole slide section. Further, there's a magnification tool, which is this little dot here that I'm pointing to in the right-hand corner, and that shows you at high power magnification the tissue within that area. The software will allow you to take a snapshot, so you can take a snapshot of this image from that entire slide, and you can send that to an investigator, or you can scan the entire thing and send that. Additionally, you can open multiple images in your toolbar on the side over here. So moving on to the nuanced multispectral imaging. So the nuanced microscope has a camera that has flexible bandwidth. And what this means, it will allow you to do both bright field and fluorescent microscopy. So you can have chromogenic or fluorescent-based images. There is a filter within the camera that switches the bandwidths and allows you to detect common markers, chromogens, fluorophores. The software provided with Nuance allows you to build a spectral library for each chromogen or fluorochrome that you're using, and as such will allow you to unmix the images, and by unmixing you're able to quantify downstream. And in addition to being able to utilize the Nuance software, the images that are provided or generated from this camera can also be used in the Inform software. So again, multispectral, not multiplexing, but multispectral. Why would you want to do that? So as I had indicated previously, investigators used to stain serial sections, one section with one marker, the next section with the next marker, and try and line those up and get a spatial relationship. There's no longer the need to do that. So nuance in a single tissue section can allow you to visualize a nuclear, a cytoplasmic, a membrane marker within the same section, and you can have multiple markers within the same compartment can be identified. So again, cell-by-cell expression of various markers can be appreciated, and this is captured quantitatively. So nuance has both bright field and fluorescent capability. Here is the spectral range, 420 to 720 nanometers. The analysis capability within nuance allows you to identify pixel-based regions of interest, to do co-localization, to compare images, and batch process. And the images are in a format that allows you to do quantification in Inform, which Wei will talk about very shortly. So as I stated, this will allow you to detect even weakly expressed antigens and those that are overlapping in fixed tissue sections or fixed cells on a slide. Up to 5 or 6 markers can be run in one section. The filter sets that we currently have available through the TRIP laboratory are Bright Field, DAPI, FITSI, Psi3 or TRITSI, and Psi5. And the software also provides for autofluorescence removal, which can be an issue for those investigators that are using fluorophores. And so images can also be used in Photoshop, programs like ImageJ and Inform. So what does multispectral mean? So we all know that on a single tissue section, there's an X dimension and a Y dimension. So it's a two-dimensional image. But there's also a spectral dimension that corresponds with the transmittance and the wavelength. And so if we take one pixel, for example, and follow it through the various spectral dimensions, we would have different transmittance and wavelength. And what that correlates to, what we would see visually, is this chromogenic or this spectral library that's generated for each fluor or each chromogen. And as you can see here, there's 4 different markers that they were investigating, and each one has its own characteristic spectrum. And this is what's used to separate those images and then quantify. So this is an example that's provided by the manufacturer. Up in the left-hand corner is the image that we would see with our own 2 eyes. So this is a red, green, blue image. And then this is an unmixed, or based on the spectral library. This is unmixed into its individual channels for each fluorophore. Autofluorescence is shown here in the corner. And the unmixed image, so the composite image that's generated by the computer software, looks like what we see here on the bottom. And the manufacturer, as you know, will always provide beautiful images, and you ask yourself, can this really happen? Does this really happen? And so this is from an investigator's laboratory here on campus, and you can see here very nicely. This is the red, green, blue, the RGB image. When the spectral library is created and the specimen or the image is unmixed into its individual cubes for each fluor, and the unmixed composite image is generated, you can see that it is very similar to that which the manufacturer provided. This works very, very nicely. So some helpful hints on using fluorophores. So many of those who use them know that it's best to use those that are more photoresistant. And when using multiple in a single tissue section, and you know that you may want to do some image analysis downstream, the best results we have found is if you have at least 50 nanometers between your respective fluors that you're trying to separate. And here is a table that the manufacturer has provided. This is available on their website. You can contact us. We can provide this as well when you're getting ready to do your studies. For QDOTs, Alexa Fluor, the various filter sets that they recommend. chromogens that the TRIP laboratory has worked with in the past. The two vendors are BioCare Medical and Ventana, and listed below are the names of the chromogens that have been used from the respective vendors. So using Nuance software, one of the advantages is that it can do two things. It can help you separate or identify chromogens that visually look very similar and can highlight those for you. So if we look at the left image, this has been stained with a dab and then a red marker as well. And visually, when you look at it, it's very difficult to discern which is the brown stain and which is the red stain. However, using the Nuance software and the creation of the spectral library, followed by the unmixing, it is able to separate those chromogenic spectra and then highlight for you, in this particular instance, they were interested in co-localization, so which of these particular cells have both red and brown, and that's highlighted here in the yellow. So very difficult for us perhaps to see visually, but the software very nicely is able to identify that. So a similar image where they're looking at a red and a brown, so a red stain and a dab stain. Here on the right-hand side, we have the unmixed, or the image that's created, the composite image that's created by the software. You're able to identify or choose the components that you're interested. So the red stain and the brown stain, and you select whatever color choice you would like. You set your threshold values for indicating what you think is going to be considered positive for that marker. And then in the co-localization software options, you can ask for the software to co-localize 2 items, 3 items, and then to select out which denominator you're interested in. Is it just the red stain? Is it just the dab stain? Is it all of the cells that are within the image? And that is printed out for you within the statistical portion that you would select here. So here is a working example of just that. So this image is taken. The red is denoted in red. The DAB, or the brown, is denoted here in green. And then the counter stain hematoxylin in blue. Threshold levels have been set, and then co-localization has been selected for those markers that are red and brown, or dab. In this instance is green. And we select that we want the color, when co-localized, to show up as yellow. And so the software very nicely tells us that about 6% of the cells in this particular image have co-localization. And then it breaks it down further, the number percent within the red and the dab. Additionally, you can change what your denominator is. If you select multiple options, it will give you multiple statistical analyses at the bottom based on what you have selected. So in this instance, we're looking at those that are overlapping versus those that are present within the full image. It will give you a marker area. So if you select your region of interest versus the whole image, it will tell you how many pixels. In the case of a chromogen, it will tell you the optical density, the total signal is printed out here. It will give you an average signal, the max signal, the standard deviation, etc. So this software is very powerful, but still somewhat limited, and that's why many investigators choose to go on to use the Inform software. And so I'm going to go ahead and hand it over to Wei and see if she can tell us a little bit more about what Vectra and Inform have to offer.
Thank you, Kristina. That's a very nice overview. So my part, I'm going to focus on more on practicality and the experiment we have done in the TRIP lab and how we can use it for our research purpose. It's more about application. So And so Kristina has introduced to you the nuance system. Actually, nuance system is Vectra belongs to part of Vectra system. So before I go on talk about Vectra, I'm going to talk about a little bit the background. So as we know, traditionally, in the assessment of biomarker expression is done by manual estimation of staining intensity, and commonly reported as either binary or four-tiered, or sometimes semi-quantitatively with a H-score. However, as new molecular technology advances, such as serial analysis of gene expression, cDNA microarrays have enabled measurement of thousands of genes in a single experiment, revealing hundreds of potential important candidate cancer genes. So to validate the diagnostic, prognostic, and therapeutic importance of the emerging potential cancer genes, sometimes hundreds of tissue samples from cancer patients at different stages need to be analyzed at protein level. However, the analysis oftentimes is limited by the speed of evaluation, creating a biomarker bottleneck on a microscope stage of academic pathologists, and by the inherent shortcomings of a manual quantitation. So what are the shortcomings of the manual quantitation? While the eye can be very sensitive to low light levels and can also see very bright conditions, it does not discriminate light levels very well. The eye also has a limited ability to discriminate levels of illumination ranging from the lowest level to the highest level at any given sensitivity. With the inherent limitation of human perception of light comes intra- and inter-observer variability and limited precision. So one recent advance is the development of Vectra technology. It merges automated slide scanner, multispectral imaging technology, what Kristina just described, and unique pattern recognition-based image analysis into a powerful system for biomarker discovery and clinical studies. Vectra is designed to measure protein expressions and morphometric characteristics in distinct tissue regions of interest on TMA or on whole section slides. Sections can be labeled with either immunofluorescence or conventional IHC stains, or with stains such as H&E and trichrome. Up to 200 whole section slides can be scanned in a single run. This picture shows the Vectra system. On the left side, this is a slide loader. This is a motorized stage and a nuance system, and this is a computer with software. With IF or IHC, the expression levels of single or multiple proteins or molecular markers, including mRNA and DNA, can be measured on a per tissue, per cell, and per cellular compartment basis, even if those signals are spectrally similar, spatially overlapping, or even obscured by autofluorescence. The number or size of tissue structures of interest can be counted or measured. Area of different tissue types of interest can be measured. Cells of interest can be counted as well. Usually a scanning protocol will be created based on the TMA core number and size or tissue morphology. Nuanced multispectral image cubes, either 8 bits or 12 bits, and using a full CCD frame for analysis. You can choose 8 bits or 12 bits. In general, 12 bits has a higher resolution. Digital electronic sensors are linear in response to light level that's superior to human eyes. That's why we choose to use a morphometric system. to do the analysis. So what we have is a nuance FX model. This software is used to build the spectral library, as Kristina had already showed. Each chromogen or dye has its unique spectral characteristics, which is the basis for building spectral library. Nuance FX operates on or off microscope in this range, spectral range. In theory, 14 ideal chromogens or dyes with peak spectral difference greater than 20 nanometers can be stained on a single slide and mixed for quantitation. In reality, maybe 10 can be done if you try. I think it's not impossible. Now, I often being asked, so which way to go, to use bright field or dark field fluorescent assay? Which one is better? So nuanced multispectral imaging technology enable us to study multiple markers simultaneously with either one. So can be done. Each assay has its pros and cons. So this table summarizes the differences and similarities. So for bright field, because the limitation of the chromogen available, you maybe do up to 5 markers reliably. But with fluorescent assay, since the chromogen, the spectral bandwidth is much narrower compared to chromogen. So you can maybe do more with narrow mode of nuanced software. And spectral library, chromogen, it's a chromogenic assay, it's just chromogen specific. With an IF assay, it's both dye specific and also tissue specific because each tissue has its unique autofluorescence. You have to take that into consideration. For scanning, it's a lot easier to do bright field, just one scanning for every dye, one exposure. However, for fluorescent assay, you really have to spend time to determine the optimal exposure time for all the channels for you to achieve the best result. Otherwise, some of the marker may be oversaturated while the other underexposed. And with masked antibody, for bright field, it's optional because we can see the morphology with naked eye, yet with fluorescent assay, without a masked antibody, you don't know where is your target compartment if there's no staining. So oftentimes, you are just looking at a dark image. Also, the other difference between these two assays is the fluorescent dye are mostly photosensitive. So you have to choose a fluorophore that's relatively resistant to the light. So in general, there's pros and cons for each assay, so you have to decide which one you want to go. You have to take all things considered and decide the assay to do it. This images show you, you can see this spectral library, you can see each spectrum, the bandwidth is relatively narrow. We have 4 dyes on this spectral library. The black is the autofluorescence. This picture shows the spectral library for 4 chromogens. As you can see, the bandwidth for each chromogen is much broader. That's why it's relatively limited. The narrower the bandwidth, the better resolution it is, and then you can do more markers with narrower bandwidth chromogen or dyes. So the common applications, broadly speaking, there are 4 categories we can do: biomarker quantitation, biomarker co-localization analysis, tissue segmentation and measurement, and cell structure counting. So for biomarker quantitation, you can obtain continuous data, which pretty much is just mean optical density or mean counts. If it's for bright field, it's optical density. If you are using fluorescent assay, it's the mean counts per pixel area in the tissue or cell segment of interest. Also, you can do categorical data. You can do either 2-bind, 4-bind, 10-bind, or even 50-bind data analysis. So I'm going to give you several examples that had been done in the TRIP lab. So the first example, in this experiment, we did validation of vector system with multiplex IHC assay with E-cadherin and Biomarker. These two markers are known prostate cancer markers. And also, we study AR and ERG expression levels and co-localization. AR is androgen receptor, and ERG is ETS-related genes, which is known prostate cancer marker. Also, we study AR expression levels in ERG positive and negative prostate cancer tissues. Because in general, people know that in prostate cancer field, ERG expressed in about 50% of prostate cancer patients. So I used two cohorts. One is a progression to construct two TMAs. One is a progression array, and the other is outcome array. And I stained the slides, one progression array with two antibodies, E-cadherin and Biomarker, and with counterstain, hematoxylin counterstain. And the other two TMAs, one is progression array, the other is outcome array, and stained this two TMA with a similar panel of 3 antibodies. They are E-cadherin, AR, and ERG. Just want to verify the data, the two sets of TMA data, verify each other. So this images show you the spectral library with constructed and built with these 4 dyes. Actually, yes, 4 dyes. So, and then this is the picture of the TMA, a low power view. And this is an image of the progression array stained with these 3 antibodies, AR, ERG, and E-cadherin. And this imaging just shows you an example area of this TMA, one core tissue. We unmix, you can see the 4 channels. This is AR, DABI, ERG, Wart-Red, and E-cadherin, and hematoxylin. And how are we going to do it? First, for this is just showing one core. How do we do tissue segmentation? This using the artificial intelligence pattern recognition software. It's called Inform. You draw by example. You draw this area, just tell the software this is epithelium, and this area is the stroma. You draw different area, in general 3 areas of each, and then the software can learn and just segment tissue into epithelial compartment and stroma compartment. Then you can decide which compartment you want to use for quantification or further segmentation. Now, since I'm only interested in prostate cancer, so the epithelial is the compartment I'm interested. So I'm going to further segment the epithelium into cytoplasm and the nuclear compartments. Just so you can see the membranous compartment, nuclear compartment, and cytoplasmic compartment. So Inform software offers several ways to quantitate the biomarker expression levels. Understand the tissue and the cellular localization of biomarkers that you are studying in order to determine how the analysis should be done and to achieve the best possible results. So this figure just shows you can see Biomarker is mostly expressed in the apical region of the prostate epithelium, and E-cadherin is mostly membranous, and AR and ERG are nuclear compartment. So you can see with this apical expression, when you do cell segmentation, the staining falling out of the cellular compartment. So if you choose to do either nuclear or cytoplasmic compartment for quantification, the result wouldn't be reliable. So in this instance, it would be better to use epithelial compartment to quantitate MCAR. So now, this table shows the data I acquire with epithelium compartment, shown here too, to quantitate E-cadherin and MCAR data. And I use IBM SPSS software to do the plotting. As you can see, MCAR expression, it's significantly elevated in high-grade PIN and prostate cancer compared to the benign prostate tissue. And E-cadherin is decreasing as the cancer progresses, which is consistent with published data. So this experiment verifies the vector system and the multiplexing IHC. And this table shows the subcellular quantification data. As you can see, the same experiment, I used the nuclear compartment to quantitate ERG and AR. That's how the data look like. And the plotting, you can see in two cohorts, the data, quantitative data are very similar. This blue bars shows nuclear AR expression level, and the green one shows ERG expression level. In this cohort, you can see very similar pattern, so which indicates the quantitation really is reliable. And this two figures shows, because I further stratify the cancer patient into ERG positive and ERG negative, and to quantitate AR expression level in the two categories. As you can see in these two cohorts, the data trend is very similar. So what I found is AR expression level is increased in ERG positive patients. So this is just table further showing the data. And the third experiment I did is to look at the co-localization and AR and ERG co-localization status. And you can see this 4 highlighted column. Just use this as example. You can see 7.1% double negative, and close to 0 single ERG negative, and 88% AR positive, and about 5% double positive for both AR and ERG. So experiment 2. So this experiment just to explore ER-beta as a predictive marker for breast cancer progression. So we did some experiment. As you can see, this is our TMA, breast TMA done by Trip Lab and owned by TSB. And you can see we can do this categorical quantitation. As you can see, we can quantitate ERG expression into negative, 1+, 2+, 3+ for each patient. So move on to the next category, cell counting. So we did experiment using a prostate TMA slide stained with AR, ER, and smooth muscle actin, just to see if ER play a role in cancer development. So pretty much it's the same. You train the software to recognize where epithelium is, where the stroma is, and there is a software you can do cell counting. You have to set up a threshold, which is operator defined threshold. So you can see I set up a threshold as I think 0.1 as the threshold for DAB, and then you can see the software recognize those positive for ER, the cells for ER. So AR, actually, this is one for AR, positive, because it's very few cells are positive for ER, so I used AR to do this. And here you can see, it just tells you in epithelial compartment, about 200 cells are positive for AR. This is just showing one image. If you want to, like, say, analyze 200 image, you have an Excel file to show the data. So I'm just showing you one core data, so which can be done. So next category is tissue segmentation and measurement. So in this one, we try to assess prostate atrophy associated with finasteride treatment. What I did is I used some substitute factors or parameters to estimate the atrophic status by measuring the ratio of epithelium to stroma and the ratio of luminal epithelium to the basal cells. So what we did is we stained the prostate section chips with CK5 and PSA and AR. CK5 is used to define the prostate basal cells. PSA is used to define prostate luminal epithelium, and use hematoxylin for counterstain to define the nuclei. This image shows, this is a composite image. This is a finasteride. The bottom one is a patient sample treated with finasteride. This one from patient without finasteride treatment. You can see by morphology, you can see there is a difference. And in the top one, you see more prostate epithelium. And here, there is more stroma. And you can see the dark one defined by cytokeratin CK5. That's basal cell hyperplasia. So from morphology, you can see there is a difference in the two groups. But by eyeballing it, it's very hard to really precisely quantitate that. So you can see in this one, that's prostate highlighted luminal epithelium. This one is AR, highlighted nuclei, and you can see the dark black basal cell highlighted by CK5. And here, this is hematoxylin stained image. So we did quantification. This highlighted area shows you the pixel area of epithelium in that image, and this highlighted area shows the pixel area of the stroma. And if you do, you divide the epithelium by the stroma pixel area, you got the epithelial to stroma ratio. So we plot this, as you can see, this the y-axis shows the epithelium to stroma ratio, and this x-axis shows you the treated and non-treated group. As you can see, there is a significant difference of the epithelium to stroma ratio in these two groups of patients. And that's with nuance informed software. However, if you want to do the the luminal epithelium to basal cell ratio, because the pattern recognition software is not that cannot really just can do sort of a macro segmentation, cannot pinpoint that the resolution isn't as high as you want to really segment basal cell from luminal epithelium. So you have to use nuance to do this, as Kristina already showed. So you can do that. You can just threshold in both staining, and you can get the size, pixel size of luminal epithelium by color, and also the basal cell epithelium are the same, just like a epithelium to stroma ratio. And you can do that. You get the measurement, and you can plot, and you can see the non-treated group versus the treated group, there is a significant differences. So which proves our hypothesis that there is a significant difference after finasteride treatment. There is a atrophic change in the treated group. So also you can do with same set of image, you can measure PSA level in epithelium, in stroma, in different groups, and also you can measure AR, and you can measure even the stroma AR level. So you can get a lot of data from a single experiment. So in summary, Vectra is a powerful system for automated morphometric analysis of tissue cell samples. It is high throughput and precise, generating continuous and categorical data at cellular and subcellular levels, and can be used for co-localization study, and it can do cell and structure counting. However, there's always caveats. You have to know the distribution and the localization of biomarker of interest, and to understand the limitation of vector system, and design your experiment well and select appropriate forms of analysis to generate and maximize reliable data. So if you decide to use this system, you may choose to do the analysis yourself or find a collaborating pathologist to do it for you. You may contact a Trip Lab by email, by phone, and we can provide project consultation, training for using Nuance and Inform software, and the fee is affordable in general. So if you want to know more about the vector application, so there is a publication, then this is our contact info. Thank you. If you have any questions.